After studying this module, you shall be able to know how proteins are engineered, learn which properties of enzymes can be modified, and which are the proteins which have been engineered so far. Let's learn one term before we proceed ahead, which is directed evolution. Using modern day molecular biology tools, one can isolate a gene for a protein and express it in some another host organism. If one in this particular procedure modifies these genes and expresses it in the way the original genes are expressed, the modified proteins which are being expressed now will have different properties than the original protein. So what we are doing basically is modifying the genes so that the protein can also be modified. We can extract out a gene from our organism, express it in the other host organism with modified or different better desirable properties. Thus, it can be used to change the properties of a desired protein or enzyme, and hence the protein or the enzyme with desired and improved characteristics is obtained that finds a better place in industrial and therapeutic applications. We are going to basically study many of such examples. This is what is known as directed evolution. We are having a direction for the betterment of these enzymes. This module discusses various ways how this can be achieved. For example, lysine. It's a protease. It's basically a serine protease which is used in detergents that we use. It is basically to digest of any of the protein stains which are present on our clothes. So it gets inactivated in the presence of bleach. Remember, bleach is a strong oxidizing agent. And because of this presence of bleach, what is going to happen is that the methionine at 222 position of lysine gets oxidized because of the presence of bleach, which is a strong oxidizing agent. And hence, what is going to happen is the enzyme gets inactivated. So lysine, which is to digest of the protein stains, is not going to act. So the gene encoding for lysine has been engineered so that this codon 222 can be, rather than having a methionine at that place, it can be replaced with, let's say, alanine. The modified protein stays functional in the presence of bleach because we have replaced this methionine now. Let's learn about how this substitution is done, how proteins with improved properties are designed. So this module answers all these questions along with the success stories in many areas with these engineered proteins. Let's learn about mutagenesis now. Directed mutagenesis. Proteins are outcome of what is encoded in the DNA. Whenever a protein is coming, there has to be a source, DNA, then the mRNA, and then the protein. So when a protein with improved properties is desired, it is better to change the protein at the level of a gene that encodes it. Prerequisite to gene manipulation, one should know which codons can basically be modified. Computer programs help us in this task. They predict the folding of a protein and ascribe the functions to that, yeah, this domain has this particular function, this motif has this function of the protein. So we can know these are the areas which are essential for the protein. This is a particular amino acid which is going to give us trouble in particular set of conditions. So they deduce which amino acids form those domains and motifs which are responsible for a particular function. If one changes or deletes or even adds specific codons, it is termed as what is known as directed mutagenesis. Once directed mutagenesis is performed for a protein, The protein which is now obtained is then checked for acquisition of desired property. So the new protein we have will have slight changes from the original protein, but is going to have the properties we want it to have. So mutagenesis can even be directed or it can even be random. We are going to study a few examples of random mutagenesis and specific mutagenesis or directed mutagenesis. The first example which is cited here is site-specific mutagenesis, which means it has to be directed mutagenesis with M13 DNA. What happens is, earlier this M13 virus exists in it basically has a single stranded form of DNA and also the double stranded form of DNA. Earlier what happened was M13 double stranded form, which is known as the Fachmet, was used to perform the directed mutagenesis. This was known as the Kunkel's method of mutagenesis. But in recent times, M13 DNA is used in its single stranded form. This M13 plus strand, which is the single stranded form of M13, is taken with the gene to be mutated. It is basically mixed with the oligonucleotides which are harboring a stretch of DNA with one of the mismatched nucleotide at one location. Now this mismatched nucleotide at one location is there in the oligonucleotide. In this figure we can see that this TTA codes for lysine. Now we want it to be modified to be 2 into isoleucine. So it can be modified to ATA, which is the codon for isoleucine. The oligonucleotide then binds to the recombinant M13 strand perfectly except at one nucleotide, which is the mismatched nucleotide where we want the change to happen. This acts as a primer for DNA synthesis. It is going to provide the free 3' OH for the synthesis of the complete DNA. And the M13 plus strand which we have taken, which is the single stranded DNA form of M13, basically serves as the template. And double stranded DNA which is now getting synthesized, these DNA also carrying, remember, the mutated nucleotide. We can see in this figure it is having the mutated nucleotide, this double stranded DNA. This is then transformed into the E. coli. Now M13 virus particles are produced from these transformed bacteria. Now since it is also having a proper strand with unmodified or unmutated sequence, so what is going to happen is if 100 virus particles are released, half of the virus particles, let's say 15 number, will be with the unmutated sequence and other half will be with the mutated sequence. So the mutated ones are identified. The double-stranded mutated DNA is used for expression of mutated proteins. We can see in this figure how this modification has been made. M13 is serving as the template. This is that shown in red is the mismatched nucleotide. This is the oligonucleotide harboring this mismatch. This mismatched nucleotide which is being harbored by this oligonucleotide is basically going to check for the change in DNA and then the change in the protein which we require. So this is the usual DNA synthesis which is occurring. Clenofragment is given, DNTPs are there. So it is going to form the double-stranded DNA with one base mismatch. Ligase is used to seal the nick, transformed into E. coli, and then various M13 plaques are produced. Now these plaques are going to have the unmutated and the mutated sequences. Half are going to have the mutated ones, half are going to have the unmutated ones. So this is how site-directed mutagenesis is performed using oligonucleotides with the mismatch. Let's look at another example how site-directed mutagenesis can be performed. It is PCR-based site-directed mutagenesis. Now the method of site-directed mutagenesis is to basically make point mutations, even switch the amino acid. It can be even used to delete or insert single or multiple amino acids. The enzyme which is used is PFU DNA polymerase. You must have heard about clenofragment or tag DNA polymerase. Those two are not used in this particular case because this PFU DNA polymerase basically has high fidelity and extremely low error rate, which is what is required. Because when you are conducting a PCR reaction, when you are carrying out a PCR reaction, you do not want that the secondary mutation should come. The base you want to modify should only get modified and the other bases should remain untouched. So this PFU, which is isolated from pyrococcus, this PFU DNA polymerase is used in this reaction, and what else we require is a PCR machine in which we can have 3 different temperature setup. So the basic procedure, which is basically developed by Stratagene, which is now called the Agilent, utilizes a supercoiled double-stranded DNA. Now this double-stranded DNA vector is there with the insert of interest, and we can see in this figure that the 2 synthetic oligonucleotide primers which are carrying the mutation at the desired site are also added to this PCR tube. So your PCR tube has the template now, which is the double-stranded DNA. It has 2 oligonucleotide primers. It has the PFU DNA polymerase. We are also going to put in various other chemicals which are required to conduct the PCR reaction. So the oligonucleotide primers, let's learn about how they are designed. Each is complementary to the opposite strands of the vector. They are extended during the temperature cycling by PFU DNA polymerase. Now, incorporation of the oligonucleotide primers generates a mutated plasmid containing these staggered nicks. After we are done with the PCR reaction, after the temperature cycling per se is over, the product is treated with an enzyme, a restriction enzyme which is known as DPN1. This DPN1 endonuclease, whose target sequence is GATC, you require the methylation of this particular sequence GATC. So it is DPN1 endonuclease is basically specific for methylated as well as the hemimethylated DNA, and is used to digest the parental DNA template and to basically have the select out the various mutation containing synthesized DNA. So what you are going to have after DPN1 treatment is that the parental or the original DNA is lost, and we are only having enriching the mutated DNA now. So DNA isolated, this DNA after DPN1 digestion is transformed into E. coli. So this DNA will be mutated DNA now. So DNA isolated from almost all E. coli strain is dimethylated, we know, and therefore susceptible to DPN1 digestion. So the nicked vector DNA containing all the desired mutation is then transformed into the bacterial cells after DPN1 digestion, which is basically going to enrich the mutated DNA. We can see in this figure how mutant strand synthesis is carried out. This is the original parental DNA. These are the oligonucleotide primers. These red dots show where these mutations are there. So this is the mutated DNA synthesized after the PCR reaction. Remember, there are no secondary mutations in this. Mutation is only present where we require it. So after this, it is given for DPN1 digestion, which is going to remove off the original or the unmutated DNA. The PCR amplified DNA cannot be methylated until and unless it is given into a E. coli system. So only the parental DNA, which is methylated or in a hemimethylated state, will get digested off, and we are only going to have the enrichment of the unmethylated or the mutated DNA now. Once it is transformed into the competent cells, for the nick repair, and that is how we enrich our mutated DNA. This was with the specific or directed mutagenesis. There is also a way how we can mutate our DNA, which is random mutagenesis, which is by error-prone PCR. In case of PFU, we made sure that there are no secondary mutations which are happening. In this case, we are allowing it so that the PCR is more of error in this particular PCR. By now know that tag DNA polymerase bears low fidelity than the PFU DNA polymerase. Now this fidelity can be further lowered if we have the presence of manganese rather than the magnesium, which is in general taken. So first change we have made is to have manganese instead of magnesium chloride. The second change that we have made is to have the tag DNA polymerase rather than the PFU DNA polymerase, which we just studied. So also the 4 nucleotides which are being provided for the PCR are in unequal proportions. So these are the 3 changes we have made to the usual PCR reaction and conducted the PCR. PCR in these 3 set of conditions, the mutated DNA which is generated has mutations at random locations. We can see in this figure this mutated DNA, we can see this is the template DNA and the primers are there. PCR is basically error prone PCR, so these dots represent where the mutations have happened. So this is not going to basically give us one mutated DNA, it is going to provide us with a library of mutants which are then screened for the desirable change in property in the protein. So this is with the random mutagenesis, and we also learnt about the directed mutagenesis. Now let's see how these mutagenesis tools are being used for our protein engineering. Remember in previous modules we have elaborately discussed about various properties and features of various proteins and enzymes. Their properties such as catalysis, substrate specificity, pH optima, stability to various thermal and pH inactivation are the various targets for improvement. Biotechnology aims to improve the chemical properties of these enzymes so they function better in low substrate concentration. They are stable in extremes of pH and temperature. Protein engineering thus involves the use of genetic manipulations. Genetic means in the gene itself to alter the coding sequence of a gene and thus the properties of the protein encoded by that gene. So by protein engineering we can improve upon even the stability of the protein. How can this be done is by adding various disulfide bonds as engineered in lysozyme, human pancreatic RNAs, by changing the labile amino acids. We are going to learn in this module what are these labile amino acids as engineered in those in case of triose phosphate isomerase. We can even reduce the number of free sulfhydryl groups. See an example in case of beta interferon. Other properties such as increasing specificity, abolishing the cofactor requirement, decreasing the protease specificity and increasing the enzyme activity have been engineered in tissue plasminogen activators, subtilisin, one of the cases we just discussed, streptokinase, tyrosyl transfer RNA synthase. We are going to now study the various finest example of these protein engineering. Slightly about subtilisin, let's look at it in detail. Subtilisin is a serine protease which has a wide range of proteolytic activity. Remember chymotrypsin we discussed in one of the previous modules. Now this chymotrypsin studied also resembles the subtilisin. Subtilisin also has a catalytic triad of aspartate 32, histidine 64 and serine 221 moiety and has a similar mechanism of action as that of chymotrypsin. This enzyme is used as an important component in preparation of washing powders and detergent categorized as stain removers. However, one of the residues which is present in subtilisin is methionine at 222 position. It doesn't allow its usage in the presence of bleach. Bleach is a strong oxidizing agent which basically oxidizes this methionine 222. So what happens in presence of bleach because of this oxidation of methionine is that subtilisin loses 90% of its activity because of this oxidation, and because it is losing 90% of its activity, it inactivates the enzyme and is unable to remove those protein stains from the clothes. With the aim to improve the performance of subtilisin even in the presence of bleach, one thing is for sure, that this methionine 222 should be replaced. So in an attempt to do that, methionine 222 by one of the groups was replaced by other 19 amino acids. Remember, one at a time, by site-directed mutagenesis. So this table basically represents how what those people obtained as a result from these 19 substitutions. Replacement with cysteine gave us 138% relative specific activity, keeping the 100% activity with the methionine at 222 position. But like methionine, remember cysteine also possesses a S group. So this cysteine is also prone to oxidation. The next in the list is a replacement with alanine, which though had only 53% specific activity, but unaffected by the presence of bleach. So this substitution imparted the stability to the enzyme in the presence of strong oxidizing agent like bleach. So this is one of the successes of enzyme or protein engineering field. We can see in this figure A and B that the relative specific activity with these substitutions, with cysteine it is 138%, with alanine it is 53%. However, the time of exposure in the presence of bleach basically is not hampering the activity if this methionine 222 is being replaced by alanine or serine. So it possesses whatever activity it had, it is not inactivating the enzyme. So this is relative specific activity of subtilisin and its various mutants in the presence of a bleach. So replacing methionine with alanine helped us to improve its activity and let it function in the presence of bleach. So this is how protein engineering field has helped in this particular area. Stability to a protein can be incurred even by increasing the hydrogen bonds, electrostatic interaction, which are basically stabilizing forces. Substitution of asparagine 218 to serine made subtilisin even more thermostable. Another effort was to make subtilisin independent of cofactor requirement. Subtilisin requires calcium for its activity. This requirement poses a problem when metachelators, which are basically present in water softeners, are present. So when these metal chelators are present, these are going to chelate off the calcium, and again subtilisin won't be able to function in the absence of calcium. So thus random mutagenesis can be used to abolish the cofactor dependence by deleting the calcium binding site and selecting the stable mutants from a panel of or a library of random mutants which are generated. Let's understand it with more clarity. This is the subtilisin enzyme molecule having the high activity because of the presence of a calcium binding site. Now what we have done is we have deleted off this calcium binding site, and this enzyme now with the deleted calcium and binding site possesses literally no activity. So in this what we have done is we have given it for allowed random mutagenesis. Remember we learnt about error prone PCR. So in this case we are allowing secondary mutations to happen. So these stars are in this case showing the secondary mutations which came because of random mutagenesis, and these are possessing now better activity than this molecule. Now all three mutations can be pooled into one to have a final molecule with high activity. So this is to make a subtilisin molecule calcium independent. Other success stories in the field are thermostable T4 lysozyme. The thermostability is contributed by disulfide linkages. One of the groups worked on improving the stability of T4 lysozyme by increasing the number of disulfide linkages. How they did it was they did so by substitution of few amino acid by cysteine residues. So they created 1 to 3 disulfide linkages, and the T4 lysozyme, which is now the modified one, which is now created, which is modified one, has better stability at higher temperatures because of these extra disulfide linkages which are there. Only consideration while doing this is that the two cysteines which are participating in disulfide bond formation should be close to each other in the quaternary structure and not in the primary structure. Another example is triose phosphate isomerase. The proteins with lot of asparagines or glutamine are thermolabile. Why? Because at elevated temperatures, these amino acids are going to get converted into aspartate and glutamate and release of ammonia. So this triose phosphate isomerase has many asparagine residues. The non-essential asparagine residues can be substituted without compromising on the quaternary structure of the enzyme. So now we have a modified TIM enzyme, which is our thermostable enzyme. This table basically lists various other examples of protein engineering field, how it has helped in various areas. To summarize, enzymes or proteins can be engineered by site-directed or random mutagenesis to create modified enzymes or proteins with desirable improved characteristics. Successes in this field have been achieved with subtilisin, which forms a important part of the detergents. Subtilisin is a protease which gets inactivated in the presence of strong oxidizing agent owing to the methionine that gets oxidized. Protein engineering has been used to modify this methionine to alanine that remains stable in the presence of oxidizing agent. So the protein with alanine is stable. Other successes have been in increasing the thermostability of enzymes, increasing the stability of enzymes at extremes of pH, changing their substrate specificity. So protein engineering for sure requires our prior knowledge about the 4 levels of protein structure.